Okay. All right. So, a second. One. All right. One second. Let me check. If I use this.
أما فاتورة.
All right, so we're going to be talking about like biotechnology, right? So biotechnology and like genetic engineering, stuff like that. All right, so Frihas, so what is genetic engineering? Do you know, like, have you heard of that term, or like, what do you think that is?
Construction of genes.
Something to do with genes, right? So something about constructing genes. All right, so Anas, what do you think? What do you think is genetic engineering?
Like building up a gene,
Like doing what to a gene?
like building it up and make it a human.
All right, building up genes. All right,
Modifying it.
Making a human? Now you good?
all right, Yusuf, what do you think?
You get the
Whether
idea.
engineering or making genes
Yeah,
or modifying genes.
Yeah. So basically, so in a plant or in an animal or a human being, right? There's like genes, right? There's like DNA. There's like genes, right? So genetic engineering is basically just like modifying it, right? Using different kinds of techniques in the laboratory to modify the genes, add new genes to something, right? Maybe so. In this case, take a look at this picture over here, right? You see this cat here? So these cats are like glowing, right? See the cat on the right is like green, it's like glowing green, it's like glow in the dark.
Mm.
And then the one on the left, they made this glow in the dark red cat, right? So how did they do that? Anyone have any ideas? Like how did they do that?
Well, brother, genetic modification, brother.
Yeah, so they probably took a gene from somewhere else, right? So there are creatures that do glow, right? Right? Like jellyfish and different kinds of fish in the ocean, different kinds of, bacteria, all different kinds of things, right? They glow or they produce light, right? So they took one of those genes and they put it in a cat, and so now the cat is going to glow as well, right? So this is like an example of genetic engineering, right? All this weird kinds of stuff these guys are doing, right?
Mm.
How do they get?
So we're gonna talk a little bit about how they got the gene and how they put it in a cat and stuff like that, right? All right. So what is genetic engineering, right? So genetic engineers are basically like scientists, and they use technology, right? Microscopes. They use laboratory equipment, and they analyze and even change the genetic makeup of living cells. So they take one cell and they can actually put in new kinds of DNA, take DNA out, right? Stuff like that. So, this picture is just kind of describing some things that they do. It says genetic engineering is the direct modification of an organism's genome. So the genome is basically like all the chromosomes in an animal or a plant or whatever, or in an organism. So modifying it, right, like taking a gene out or putting a new gene in, different things like that. This is all genetic engineering. So people use this for like different kinds of purposes. Anyone know like any kinds of purposes or reasons why people do genetic engineering? Like apart from just for fun, right? Why would people want to do genetic engineering?
It's used.
To cure cancer.
To cure cancer? All right, Yusuf, what do you think?
It's used in food a lot because like to modify it, make it bigger, I guess, or make it produce more, become like a faster growth rate for people who are like deprived of food that don't have much food supply.
Right, that's correct, right? So like, for example, like people have been like crossing different kinds of plants for a long time to produce, fruits and vegetables that are like bigger and stuff like that, right? Like you might start with like some small thing and turn into like a big orange that has like a lot of juice.
Like more efficient, basically, right?
Right. Yes. For our purposes, like more efficient, right? It produces, like one tree will produce like more of whatever we want, right? All right, Freeha, what else? What
Or
kind
one fruit gives a lot of benefit.
of? So Freeha, what else? Why might people use genetic engineering?
For food.
Yeah, for food, right? So those are some reasons, right? There's other kinds of reasons as well. So for example, in plants, right? There's any kind of insect or fungus could come in and, destroy your crops, right? So they might take some kind of gene that produces a protein that kills insects, and they might put it directly into the plant. So instead of spraying something from the outside, right, some kind of insecticide from the outside, the plant itself is now going to produce some kind of chemical or protein that kills insects. So that's a way to stop insects from eating that plant, right? All right. So let's take a look at some concepts first about how genetic engineering works. So here's an example of something, one technique that genetic engineers use. So if we take a look at this, sequence, right? So look at the first row, right? You look at the first row. So the first row is basically like two fragments of DNA, right? You have the one on the left and the one on the right. And so what they can do is that there are certain kinds of enzymes. They're called restriction enzymes. And so these enzymes, they cut DNA in specific places. They're able to read the DNA, and when they get to only like a specific kind of sequence, like each enzyme, it recognizes a specific sequence. Like maybe one enzyme recognizes like A, T, G, G, C, something like that, right? Another one recognizes like C, C, T, A, something like that, right? Each one of these enzymes recognizes like a different kind of sequence. So as it's going through the DNA, it travels through the DNA. When it gets to like its specific sequence, it cuts it right there, right? It cuts it. So if you go to two different pieces of DNA, two different kinds of DNA, right? And you cut each of them there, then you can take those two pieces of DNA and put them together and then fuse them together, right? So let's take a look. So I think on the next page, let's take a look at like an example, right? So for example, let's take a look at this picture at the bottom, right? You guys see this picture at the bottom with just the white. There's no blue here, right? So you have a restriction enzyme that can read this sequence. You see this G, A, T, T, C, right? So whenever it gets there, it always cuts it in this fashion. You see the two strands are now cut like this. You guys see that? It's like now cut like this. Now, what do you notice? You notice on each of them you have this AATT part where there's nothing, there's no like corresponding, DNA sequence right next to it, right? You see how they cut it so that this end right over here is just kind of hanging, by itself, right? See AATT. Each of these ends is like kind of sticking by itself, right?
Yeah.
So it needs like another, side to attach to, right? Because DNA wants to be double-stranded.
Oh.
So you see this kind of like one-stranded portion on each of these, cut ends. Those are called sticky.
Yeah.
Those are called sticky ends, right? Those are called sticky ends. And so what you do is you take some kind of gene, right? Now we go to the top picture. You take whatever kind of gene that you want to put into the DNA, right? And then you also cut it with the same. Sequence. And so now these two sticky ends can now attach together. And so you can kind of cut and paste this gene into the DNA.
But then, brother, it doesn't change anything. I mean, I guess you get the same thing, right?
No. So this target, you see this thing that says target gene in the blue? This came from somewhere else.
But it's the same like genetic. It's like the same base sequence, though. So like, how does that make a difference?
No, no. So you see in the purple it's the same, right? You see that?
Mm-hmm.
This purple end is the same, and this purple end is the same, right? That's the same as what was down here before.
Uh-huh.
But you see this part in the
Exactly.
blue. You see this middle part in the blue.
Yeah.
This is presumably like some new other kind of gene, right?
But the sticky end is the part where
Yeah.
the
So the
sticky end is the part where the AATT is. So how is there stuff in between?
right. No, okay.
How do they put stuff in between?
All right. So this came from somewhere else. So this right over here is something that either they built somehow, or it comes from another organism.
Yeah, but how do they put it like in there? Like, how do they put it in between these two base sequences? Basically, is what I'm saying.
So
Because the sticky ends for the purple things are AATT. So how would they like put it in between?
right. So I don't remember like exactly how they built it. Let me see if we can figure it out. I don't recall how they built it. Let me see.
No, I'm not saying like how they built the part. I'm saying like how did they put it in here?
Let me see. I don't remember. It could be just that. It could be that they select. Let me see. So I think one thing is what they do is they select the restriction enzymes.
Mhm.
So I think one thing that they do is they select the enzymes such that there's kind of like a coincidence, right? So okay, so let's say this target gene came from like another bacteria, right?
Oh.
So they go to that bacteria and they figure out, okay, what kind of enzyme do I need to cut this gene out?
Oh, all right.
Because they look to the left and they look to the right and see, okay, what kind of sequences are there where we can select a particular one of these restriction enzymes to cut it so that we can cut out this target DNA.
Oh, I get it now. Okay, I see. This whole part, the AATT, and all in between was cut off.
Right. What's in between is what we want to put in.
Okay, I see. So that was also cut off with those two, right?
Right. Yeah. So basically, from like, let's suppose this gene came from bacteria. Well, they cut it off over here and cut it off over here, and so what's in between comes along with it.
Oh, okay. I didn't know. I didn't see that. All right.
So then once you put it, all right. So here's what happened. Okay. So this enzyme, right? So they have these enzymes called restriction enzymes, and do you see this picture at the bottom? Right. So each enzyme, cuts in a specific way. So they have this enzyme that cuts the DNA like this. Right? You see what happened here? They cuts it like this. So do you see each one of these? You see this top portion over here, AATT,
Yeah.
and then this portion over here, TTAA. You see how now? You see how now those portions are kind of like single-stranded, right? You see how usually
yeah. Okay, yeah, those parts. Yeah.
right? You see how usually DNA is double-stranded. There's two layers, all right?
Mm-hmm.
And so you've cut it so that each one is like a single layer. So you see this AATT, it wants to attach to a TTAA. Remember, A and T goes together, right?
Mm-hmm.
Right? G and C always goes together, A and T always goes together. So this end over here is called a sticky end because it wants to go and stick to another piece of DNA to form a double-stranded DNA again. So if you bring this target gene right in between these two ends, right, like this portion over here on the left, used to be this portion over here. This over here used to be this over here. And so if you bring this target DNA with this sequence, exact sequence here, you can paste it in the middle. Right, like this end over here is going to now attach to this end over here. And this end over here is going to now attach to this end over here. And so now within this plasmid thing, within this DNA, now you have this extra blue piece of DNA that's now there. So whatever cell this is now going to start expressing and making whatever this target gene is going to be making. All right, let's take a look at another picture. Let's see if maybe this can help as well. So this tells you the overall kind of thing, right? So here's what we want. You see this blue foreign DNA, right? So what we want is that we want to take like a certain gene from the blue foreign DNA, and we want to put it into the red, right?
Yeah.
So we want to take something from a cut piece from the blue and stick it into the red. Why? Because we want this red is like some kind of bacteria, and so we want this bacteria to make the blue protein, right? Whatever protein this blue is making, we want to put into here and force this red bacteria thing to make that particular protein. So what they do is they use these restriction enzymes and it cuts it, right? So it cuts the red, right? So you see now the red is now cut like this.
Yeah.
And it does the same thing with the blue. It cuts the blue. So now the blue is in all these pieces. And so you mix them all together. And so sometimes some of these blue pieces go here and stick into this red like this. And so now this blue gene is now part of this red plasmid like this. So plasmid is basically the DNA of a bacteria. So this blue thing, right, it codes for some kind of gene. Maybe it's like an insulin gene or whatever it is. And so now the bacteria that used to have the red plasmid, now it's going to be making that blue gene as well. Right? So this way, one organism can now start making a protein that came from another organism. Right? Freya, does this kind of make sense, the idea?
How many amino acids does a protein have, like approximately? Like what's like
yeah, so it
what's
just
the
depends.
lowest you could go?
So some are like less than 100, some are like thousands. So it just kind of depends.
Oh, there are like proteins that have like thousands of amino acids?
Yeah, so so scientists, this is what they do. So they're taking some DNA from one organism and sticking it into another one, so that one of these organisms is making the proteins from the other one.
This is just like the basic idea. I'm very sure it's more complicated, right?
Right. That's the basic idea, right? So there's other ways, like for example, like how do you verify that this, bacteria is now actually making the protein and stuff like that, and it got attached properly. So all those are like additional steps. All those are like additional, steps. But this is the basic idea, right? All right. So when you do this, right? When we do this, these organisms that are now producing DNA from some other organism, these are called transgenic organisms, right? So for example, here's what's going on, right? Let's start with the number one. You're identifying some gene, right? Like for example, let's suppose we want some plants to start making a gene that makes, a protein that kills insects, right? Because we want to grow some kind of crops, and there's too many insects coming and eating the crops. So we want those plants to start making proteins that kill insects. All right. So we identify what protein that is. All right. We cut it out and we insert it into a plant cell. All right. So then we grow the plant cell, and eventually it becomes like a plant. And so now in number 3, these plants that are growing, they now make this protein that kills insects. And that protein that kills insects, it came from somewhere else. Maybe it came from a bacteria, maybe it came from a different plant, maybe it came from an animal, it came from somewhere else, but now this plant is making that protein. All right? So that's called genetically modified organisms. Like if you see on certain kinds of foods, right? What does it say? Like this says GMO or non-GMO, stuff like that. So if it's non-GMO, then they didn't do this. It's just like the natural plant. If it says GMO, then they did do some of this stuff. So like I think like probably like most plants, like most crops are now like GMO to some extent. Right?
Yeah, I think corn is like you can't find really like any corn seed without any genetically modified parts in it,
Yeah,
right?
right, exactly. Right. So I think corn is like very highly in that category. Right? So this is just like
corn is like the highest.
So this is just an example, right? So this is talking about a protein known as the Bt toxin, right? So this picture shows that this toxin came from this bacteria. You see Bacillus, whatever this says, thuringiensis or whatever it is. So this bacteria produces this protein. So they took the DNA from the bacteria and put it into the corn plant. And so now when this insect comes along and tries to eat the corn plant, it's going to be eating this Bt toxin protein, and the insect is going to die. All right? So the idea is that now, like these crops, some of these crops, they don't need to be sprayed with as many pesticides as before, right? So it's kind of like the plant is making its own pesticide. Does that kind of make sense?
Right.
Yeah. All right. So this one over here is kind of, saying something similar, right? So apart from insects, right, there's also different kinds of viruses and fungus that can also infect plants, right? So this one is saying that, scientists are also working on plants that can resist certain kinds of common viruses, like plant viruses. All right, so another reason, like I think someone had mentioned, was that, let's suppose you want to make certain kinds of food like healthier in a certain kind of way. So they made this thing called golden rice. So usually when you look at, let's say you go to the store and you want to buy something that has grains in it, right? Right, like wheat, rice, stuff like that. Usually it says something like, oh, this is like enriched flour, right? Something like that.
Yeah.
Right. What does that mean? Anyone know what that means? If it says enriched flour?
More nutrients. They just add stuff into it, like vitamins and stuff added.
Right. So basically, what they do is they add certain kinds of vitamins, usually like B vitamins or something.
That's
like
unhealthy, though. That's not good, right?
It depends.
It's not added proportionally, is it?
Right. So I think they probably. So I'm not sure what how much they put in. So like what happened was that maybe like 100 years ago, like there were in certain like rural communities, like farm communities and stuff like that, like people were eating, like basically, people were not getting like fruits and vegetables and stuff like that. So they started adding like vitamins to the grains because everybody was eating grains, like rice, wheat. Everybody was eating these basic things. So they started adding these like some vitamins to these grains. So usually that's what it is, right? So still now in some parts of the world, right, people are just eating mostly grains, right? Just like the basic kind of things that they're eating, right? So what they did is that, so one thing is you could add vitamins to the outside, like however, like whatever powdered vitamins or whatever it is, right? You could add it from the outside. Another way is to genetically modify the rice to make its own vitamins. So that this is something called golden rice. Right?
Golden rice.
So they add something called beta carotene, which is vitamin A. It's another name for vitamin A. And so this rice makes its own vitamin A. Right? So this is called golden rice. So this is just kind of showing like kind of what they, some examples of what they did, right? So from this bacteria, I think this bacteria produces some vitamin A. So they took that gene and they put it into certain kinds of plants, and they grow the plants, and now the plants make vitamin A. right?
Hmm. Oh wow.
All right, so even cows are now genetically modified. So
Yeah, that's like everybody knows that.
so sometimes the cows are genetically modified, and then sometimes they're just injected with
The
hormones
hormone.
that are produced through genetic modification. So certain kinds of hormones increase the milk production in a cow, and so this is what they do. So this is just like another example. So in agriculture, this is like there's a lot of genetic engineering in agriculture, right?
Of the
Now this one is
increasing
talking
population.
about animals are genetically engineered to produce leaner meat or higher
This one's
amounts of.
really weird, but if you read the second part,
What happened? Yeah,
this one's really weird if you read the second part.
yeah. So the first part is saying that to make the meat leaner or to increase certain kinds of nutrients like omega-3 fatty acids, they modify the animals with DNA. Now the second portion, right? So Yusuf, what is the second portion telling us?
Basically scientists added spider genes to goat DNA. So the goats produced silk fibers in their milk when they're milked.
Mm-hmm.
Those fibers you can use them to like make strings and stuff.
Right.
So it's pretty weird.
Yeah. So they
Tennis racket
so I think
chains.
silk comes from like the silk worm, and so they took that DNA
Yeah,
and put it in the goat, and so the goat is now making silk. So
but it said from spiders, so I guess spider webs. I don't know.
no, not from. Wait a
Weird.
second.
It says spider genes.
Spider
Look, it says
genes.
scientists have added spider genes to goat
Oh,
DNA.
okay. Yeah, okay. So I think
Yeah,
that's
that's
I think that's
weird.
different from the silk, or I don't know. I'm not sure. Maybe it is the same.
Yeah.
Yeah.
Yeah, I don't know. Maybe like it combines somehow it's the goat and just produces silk instead of
Right.
webs.
Yeah. So anyways, pretty strange, right? But anyways,
We can make real life Spider-Man, brother.
yeah, you could. So the next one is so a lot of jellyfish, they produce this protein called GFP. So it's like a fluorescent protein. So this protein like glows, right? So jellyfish use it for different reasons, like to attract prey and stuff like that deep in the ocean, right? So and it's used for many different kinds of things. But for example, they put like one of these GFP genes inside of a frog, and so now you see how this frog is like glowing, right? So this frog can also its skin can also start to glow, right? Another thing that they use this for is for medical research. So what they do is that sometimes you see this GFP gene, they attach it to a gene that they want to insert into a cell. And so if they put it in and the cell like glows, then that could mean that yes, we successfully put these genes into this cell. So they can select for and take only those cells that glow and get rid of all the other cells.
Oh,
Does that kind of make sense? So
Yeah.
all different kinds of purposes, that people could use this stuff for. All right. All right. Now another thing that people use this for is to produce medicine. So for example, let's take insulin, right? Anyone know, Freeha? Do you know what is insulin used for? Like, what is insulin?
It's like people use it to. Is there sugar in it?
Yeah, something to do with sugar. All right, Anas, do you know what is insulin or what is it used for?
It's like a medicine which people use for like diabetes.
Right, for diabetes, right? So you guys are correct. So when people's sugar gets too high in their blood, right, they can't control the sugar, then sometimes if the medicines don't work, then you have to give insulin. So if you want to produce like a lot of insulin in like an efficient, cheap way, one thing that you could do is you could take the insulin gene, right, from a human cell, put it into a bacteria cell, right? And bacteria grow quickly, right? It doesn't take a long time for bacteria to grow. So bacteria can grow quickly, and quickly they can produce like a lot of insulin, right? Like microorganisms, they grow really quickly. So you put it into a bacteria, the bacteria grows, and as it's growing, it produces a lot of insulin. You take the insulin out, and now you have cheap insulin, right? So other kinds of things, there's mentioning human growth hormone, blood clotting factors, cancer fighting medicines, many different kinds of medicines and things, right? If you want to make it quickly and cheaply, put it into bacteria, and then they can make this thing very quickly. All right, so cloning, right? All right, so Yusuf, so what is cloning? What do you know about cloning?
But there's like creating a copy of one organism and you're making like basically one other copy of that organism.
Right. So usually, right, like for most mammals and stuff like that, right? Usually it requires like a mother and a father, right? And then the DNA mixed together, right? So the child doesn't have like an exact copy of like the mother's DNA or exact copy of the father's DNA, right? You have like kind of like a mix of like 50% from one parent and the other. So what is cloning? Cloning is if you take an organism and then you duplicate its DNA exactly. So this organism doesn't have, like a mother and a father, but they just take the DNA from like the mother only and then create the child with it. So they did this with sheep, right? So they did this with sheep. So this is kind of showing like how they kind of did it, right? So you see at the top, it says a donor. This is the donor animal at the top. So they took like some kind of cell from this animal, and they took out its nucleus, right? So Anas, why are they only selecting the nucleus?
So they can probably like copy it.
Right. So what's in the nucleus? Why are they interested in the nucleus?
I don't know.
DNA, brother.
DNA.
The DNA, right? So they're interested in just the DNA. So if you just take the nucleus, that just has the DNA, right? So now they take an egg cell, right, from this other animal. So the egg cell is important because the egg, that's what produces a new organism, right? That's what can grow into new organism. So they take the nucleus of this cell out completely, and they put in this other nucleus into this cell. So now you have this cell here where the nucleus came from one animal and the rest of the cell came from a second animal. And then if you put it back into like the mother and then it grows, right? You notice this organism that results, all of its DNA 100% came from this one animal up here. Right? So this is called cloning.
It's weird, brother.
All right, so people are doing all kinds of strange things, but anyways. All right.
Didn't like a Chinese scientist like do it with a human successfully? Like one human, he cloned it into two.
I don't know. I think that there's always like some kind of rumors about like, oh, the Chinese are doing this and that. So I don't know. Who knows?
No, like I heard that like this one was actually true. Like there's evidence and stuff.
Oh, really?
They actually did it. It was like pretty cool.
Is it possible? I don't know. I don't know. So now another thing that they can do is something called DNA fingerprinting, right? So what is this? This is like if there's like a crime scene, right? And there's some like DNA there, right? Someone leaves like behind some hair, hair or something like that, and you want to figure out, okay, which suspect committed, did this hair come from? So here's what you can do, right? So you take the DNA. Let me see this picture. Okay. So everybody's DNA is like a little bit different, right? Everybody's DNA is a little bit different. So let's suppose you take some restriction enzymes, right? Anas, do you remember what is this restriction enzyme?
No, brother.
Remember, so a restriction enzyme.
We literally just talked about it.
So a restriction enzyme is an enzyme that cuts DNA only in specific sequences. It can read and recognize the sequence, right? Like it goes through, it reads, A, T, G, A, C, C, G, right? It goes through and reads. So it only cuts at a specific sequence. Every restriction enzyme has like a different sequence that it cuts at. So let's say we take one, right? Let's say we take one and it's called EcoR1. You see this one? It says EcoR1, right? And this one, EcoR1, it only cuts when it recognizes G, A, T, T, C. You see this? It only cuts here. right? So every time it gets to this in the DNA somewhere, it makes a cut. Then it keeps going, it keeps reading. Next time it gets to this sequence, it cuts again. Keeps going, cuts again, so on and so forth. So what's going to happen is that you're going to have a whole bunch of DNA particles, a whole bunch of pieces of DNA of different lengths, right? Like some places it's going to be short, right? And then some places it's going to be longer, right? So you're going to have a whole bunch of different sizes of DNA, right? Now, every person's DNA is like slightly different, right? So what does that mean is that if you put the restriction enzyme into a different person's DNA, right, it's going to cut at different places. Does that make sense? Right? So if you take person A and you cut its DNA wherever this sequence occurs in the DNA, it's going to have different kinds of pieces of DNA, some short, some long. You go to a different person and you do the same thing, you cut it. Same thing, they're going to have different pieces of DNA, some short, some long, right? Now what you do is then you take all these pieces of DNA and then you run them on a gel, right? So now this gel, what it does is it separates the pieces of DNA by length. So what happens is that this gel, right? This gel has like tiny microscopic holes in it, right? So let me ask you a question. Let's suppose you have some different pieces of DNA, some that are short and some that are long, right? As you run the DNA, as you push the DNA through the gel, which pieces are going to move faster? The smaller pieces or the larger pieces?
Smaller, right?
Smaller. Why do you think so?
Because they're smaller.
So they're smaller, right? So that means that they're going to go through the holes easier, right? The longer pieces, it's going to be difficult for them to go through the holes in the gel, right? And so it's going to take them a lot longer to kind of move through the gel. So look at this picture over here on the right. See how the largest pieces, they're still stuck near the top, but the smaller pieces, they were able to run quickly. And the ones in the middle are kind of in the middle. So every person, right, when you use like a restriction enzyme to cut their DNA into pieces, they're going to make different size, length of pieces. So everyone's going to have like a different set of bands, right? So if you go over here, you see how these three suspects, they all have different sized bands because the enzymes cut their DNA in different places because all their DNA is a little bit different. So then you go to the crime scene and whatever pattern that forms, you compare it with the three suspects and see, oh, suspect 2 matches. That's the person who was present there at the crime scene.
But brother, you would have to do it quickly because like if blood or something dries up, for example, then you couldn't do it, right? Because the cells or everything inside would have been dead.
So kind of. So I think the DNA, it takes a while for it to decompose. I don't know how long it takes. But
even after a cell dies, the DNA is still there?
Yeah, the DNA is still there.
Oh, okay.
Yeah. All right. So this is kind of the general idea. And this picture at the bottom is kind of telling you something, similar as well, right? So you take this DNA, right? You cut it into a bunch of pieces, right? Now everybody's DNA is going to cut into different sized pieces, right? Because everybody's DNA is a little bit different. And so what you do is you put it on the gel and you run it through. And so the smaller pieces are going to go faster. Larger pieces are going to stay near the top. All right. So then there's other steps, like you put it on the membrane like this, and then there are certain kinds of chemicals that recognize DNA, and then you take an X-ray film of it, and then you see all these bands. So I don't think you need to know all that stuff. Basically, there's different kinds of chemicals that attach the DNA and produce and they produce like a little bit of X-rays. And so
this is what forensic science is, right? It's basically just matching DNA with whatever sample you have.
Right. Yeah. So this is like a lot of what they do. Yeah. Right.
I guess also fingerprinting, like DNA fingerprinting, and then also fingerprinting, but mainly DNA fingerprinting, I guess.
Yeah, so that's why we call it
Autopsies
fingerprinting,
and
because
stuff.
it's the same kind of concept, right?
Yeah.
All right, so I think we talked about all of this here. I think this is kind of the same kind of thing. So this is just kind of showing, just more pictures just so you can visualize it, right? So you have the DNA over here on the left. You cut it up into a bunch of pieces, right? You run it on the gel, right? And it's going to separate by size, right? Now, you can't, when you separate on the gel, right, it's still invisible, right? You can't see it with your eyes, right? So the DNA is in here, but you can't see it with your eyes. It's not like it appears as blue or something like that. So you got to put some chemical on it that recognizes any kind of DNA sequence, right? And so when you do, this probe, this chemical has something attached to it. You see this little, yellow circle over here on the left? That's basically some kind of, radioactive material that produces X-rays. And so then you use then you put like X-ray paper on the gel, just like if you go to the, the X-ray machine, if someone is taking a picture of your skeleton, right? They put like an X-ray paper on the gel for like a few minutes, take it off, and develop the paper, and then on the paper you'll see where are the different bands of the DNA. Right? All right, and the picture on the right is kind of showing you the same kind of thing. I think this is basically showing you the same kind of thing. All right, and so what they're saying is that, all right, so in addition, so what you can also do is, using the same kind of method, you can figure out, relationships among individuals. So they do this for, if you want to determine their ancestry and that kind of thing, right? So if a person is related to another person, their DNA is going to be more similar. And so when you put the restriction enzymes through on it, it's going to cut it in a similar pattern versus someone who is very distantly related. So those things like Ancestry.com and stuff like that, those places where people send in their DNA, right? So this is the kind of thing that they do with it. Now, one thing here, right, that they mention is mitochondrial DNA, right? So in a cell, most of the DNA is in the nucleus, right? So the DNA in the nucleus, it comes from the mother and the father, right? But in the mitochondria, you see this mitochondria? The mitochondria also contains a tiny amount of DNA in it, right? And the mitochondria, only comes from the mother, right? So the mitochondria comes completely from the mother. So this DNA from the mitochondria, it only comes from the mother. Right? And lastly, they're just talking about wildlife. So they're saying here that, let's suppose someone, is hunting elephants or something like that, and they arrest the person and they figure out, oh, this person was hunting an elephant. They can take the elephant and test its DNA against different herds, and they can identify which specific herd this elephant came from and figure out where this person is poaching this elephant from. So this is how they're helping to fight like illegal poaching in certain kinds of national parks and stuff like that in Africa. All right, so it's the same thing. They're just talking about a different application of all these techniques. All right, so anyone have any questions about this one here? All right, Freya, any questions on anything that we talked about? No? Anas, any questions?
No, brother.
No? All right. All right, so I think inshallah, so for this one, we'll have the quiz next time. All right?
We have like 15 minutes, brother.
15 minutes. Yeah, but I didn't make the quiz for this one this time. So we'll do it next time.
All right. Fair enough. Fair enough.
All right.
Question.